Next up, we have Maeve McLaughlin. Maeve did her PhD work at Wayne State University, and she's now at Michigan State University and currently supported by an F-32. So take it away.
Thank you. Set that there. So first off, thank you to Carl for putting this together, and a big shout out to Hannah for actually doing that lovely intro into predator-prey interactions. I've studied many of the same things, and so it's sort of a perfect segue in. So just as Hannah said, bacteria are in this constant struggle to survive in their environment. Sometimes it's environmental insults, sometimes it's things trying to actively kill them. In the case that Hannah said, right, this was a Padella vibrio, this is a predatory bacteria. Sometimes these are fungi like slime molds, but the one I want to focus in on today are phage, which are these viruses that specifically infect bacteria, right? And they essentially act like molecular parasites. They will bind to their host, inject their genetic material in, begin to hijack the host machinery in order to replicate, and eventually lyse that cell in order to release those phage progeny into the environment. Now, obviously, this is not the ideal situation for the bacterial cell, and so they've developed a lot of systems to combat that, but in turn, of course, the phage also ends up developing systems to counteract those defenses. And so they're locked in this everlasting battle through evolutionary time. And sometimes this is fought with things like weapons and armor, right? So CRISPR immune systems that we hear about in the media a lot, or the curl that Hannah just showed. I always like to think of this like a duel between people with swords, right? Something more physical. But of course, just like in human battles, sometimes battles aren't just fought physically, but mentally and through manipulation. And so the reason that this occurs is when the phage infects the cell, it needs to be able to take over the host. And this often includes creating these little viral proteins that can hijack the host machinery in order to alter its transcriptional profile or metabolic state in order to enhance its ability to replicate. And in contrast to that fight between, right, people with swords, I always think of this as like a battle between wits of like Moriarty and Holmes, where they're in a chess match to outmaneuver one another. And this is something I've been really interested in, is really trying to understand these mechanisms that these phage use to manipulate these hosts. Particularly because we know that phage are being increasingly used as treatments for recalcitrant bacterial infections, a lot for antibiotic resistance, right? And if we can better understand how they manipulate their host, we can start to engineer phage to be better predators, not just for clinical use, but also agricultural use. And so to study this interaction, I've been using the alpha proteobacteria known as Caulobacter crescentus. For those who don't know, alpha proteobacteria is a broad class that's technically now changed, but we're not going to avoid that. It includes a lot of clinically and ecologically relevant species. And one of the things that I've often used it for is to study biofilm formation. So biofilms are these multicellular communities that are often encased in some sort of extracellular matrix. And we know that biofilms often contribute to disease persistence and progression. And so with Caulobacter, we can study some of the mechanisms that are really basic to a lot of biofilm formation. How do they adhere to a surface, for instance? And over on the right is this beautiful picture of this Caulobacter biofilm where you can just see that it's this mass of cells all stuck together. And of course, to study a bacteria and a phage, you have to have a phage. And the one I've been using is known as φCbK. So here's this lovely little image of it. And to the right is actually another image of several of these φCbK actually infecting a Caulobacter cell. I started studying φCbK because it was actually discovered over 50 years ago and has largely been used as a workhorse to study Caulobacter biology. But the phage's biology has basically been almost non-existently studied, except for a few pepperings here and there. And so I thought this was sort of prime real estate to start understanding this interaction between phage and their hosts. Now, when I started, our lab is actually not a phage lab. We almost exclusively worked on Caulobacter and other alphas, and that's actually sort of how the first 2.5 years of my postdoc went. I was just focusing on how Caulobacter was regulating different things. And in particular, I used some 4 genetic approaches to identify these 4 transcription factors that I've labeled here as XRE1, 2, 3, and 4. So these are known as xenobiotic responsive element transcription factors. So they have this N-terminal helix-turn-helix XRE domain where they actually bind DNA. And this is actually just a multiple sequence alignment showing the similarity between these 4 proteins, and it turns out they're actually paralogs. And through some data I'm not going to show you today, we know that these XREs act to actually promote biofilm formation. They do this by acting as a repressor of this gene known as HFIA. HFIA encodes this biofilm inhibitor protein. So by inhibiting the inhibitor, they have a positive effect on biofilm formation. I also saw that these XREs acted as repressors of the system known as GAFYZ, which was recently identified as an activator for a cell lysis pathway. And so we know that they're doing several different processes, and actually from other data, we know that they're influencing even other pathways. But as I was thinking about what I wanted to do going forward in the future, Caulobacter is great, but I wanted to sort of expand out. And so I started to look for the presence of these transcription factors in other species. And it turns out that this group of transcription factors is highly conserved across almost all alpha proteobacteria, including a lot of the ones we often think about, like animal pathogens such as Rickettsia, Brucella, or Bartonella, which are intracellular, a lot of plant pathogens like Agrobacterium, and several of these rhizobium species that are indicated with these blue boxes that are known to enhance plant growth via root nodulation and nitrogen fixation. So it was really interesting that we saw it all here, but they weren't just present in these alpha proteobacteria, but also many of the phage that infect them, and in particular, φCbK that I mentioned earlier. And we can see here, this is just, again, a multiple sequence alignment comparing the phage XRE to those host ones, and you can see that it's as similar to the hosts as they are to one another. And so this led to this really interesting question, of course, could this phage utilize this transcription factor in order to manipulate Caulobacter? Because we know that the Caulobacter can encode several copies of these to do different things. And so can the phage use it for similar reasons? The second thing I wanted to know, right, was obviously it can alter the host environment, but does this benefit the phage in any way? And so the question was, could it enhance phage infection? And of course, given the similarity between the phage transcription factor and the hosts, did the host ones have any play in this game of infection? So to start studying this, I decided to first ask whether or not this phage transcription factor could even bind to the host genome. So I did ChIP-seq analysis, and essentially this is just a plot showing on the x-axis is the genomic position in Caulobacter, and on the y-axis is the ChIP-seq enrichment. And essentially all you need to know is that each of these individual peaks is a different site in the genome where this phage transcription factor can actually bind. Turns out there's over 150 sites across the genome, and actually if we compare the binding sites from the phage transcription factor to the host ones, you see that 99% of those binding sites overlap, suggesting they're largely functioning at the same types of genes. We can also start to look at whether or not this XRE actually influenced gene expression using an RNA-seq experiment where basically we just overexpressed this phage protein in a Caulobacter and looked for which genes were differentially regulated. So on the x-axis, we have the log fold change, and on the y-axis is the FDR p-value, so the false discovery rate. And these were about 444 of the differentially regulated genes when we overexpressed this protein. You can see that most of them are repressed. And we were able to then combine this RNA-seq data with the ChIP-seq data to identify which of these were likely direct by essentially looking for one of these ChIP-seq peaks within the promoter of those genes that were being regulated. So it turns out that 130 of those 440 sites had direct binding sites for this phage XRE within their promoter regions. And amongst those genes that were being differentially regulated were two that we were super interested in, which were HFIA and GAFYZ. And so I don't have time to show it today, but we know that this XRE acts as a direct repressor of these two transcription factors. But it turns out phenotypically, if you overexpress this XRE, you get an increase in biofilm formation consistent with it acting as a repressor of the biofilm inhibitor, and actually a decrease in the amount of cell lysis that you get. And so obviously here we could tell that it was indeed affecting host transcriptional profiles, but of course this led to the second question, what benefit does this have for the phage? And to do this, I decided to look at whether or not it could enhance the phage infection, simply by asking how many phage were produced when you either didn't have these proteins or you did. And so I did some phage infection assays, and so the y-axis here is just looking at the phage per infected cell, and so this is looking at how many phage are produced during each infection. Here we have a wild-type strain with an empty vector compared to a mutant in which we deleted all 4 of the host XREs, and we can see that we get this decrease in the amount of phage that are being made. And we can actually then complement this back by adding back either the host XREs, shown here in pink, or the phage XREs, shown here in blue, and you can see that it rescues back to normal levels, suggesting that these XREs are actually enhancing the phage's infection, though the mechanism is still unknown at this point. So I want to sort of start wrapping this into a model of how we're really starting to think about this phage infection and where we're planning to go from this here on out. right? We know that this φCbK phage can infect Caulobacter, right? And during this infection, it will produce this XRE phage. And I didn't have time to show the data today, but I've done an RNA-seq experiment for an infection time course, and we can see that this phage XRE is produced at the earliest time points and very highly. And we think that this may be one way that it starts to set the cell up for the infection cycle. We know that once it's made, it can act as a repressor for HFIA and promote biofilm formation. And we think that this may actually facilitate spread of the phage through its environment by promoting these formations of these multicellular communities so that it's nearby other friends where it can then stab them in the back, right? We also saw that it acted as a repressor of GAF-YZ and inhibited cell lysis. And this is sort of an idea that makes a lot of sense, and we've seen a lot in the field of phage biology. One of the common ways that a lot of bacteria deal with phage infection and to prevent it spreading to their neighbors is by actually inducing a premature cell death. Because if you kill yourself before you make active phage particles and the cells burst open, nothing happens. So by doing this, the phage may be ensuring that those cells never enter in that premature cell lysis state to ensure they're going through a full replication. Of course, we also see that it's enhancing this phage's ability to replicate. And like I said before, this is something I'm really interested in because it's not clear at this moment how or why. But of course, we have this big data set of all these different genes that we know it's differentially regulating, both directly but also indirectly, because it could be via secondary transcription factors that it's self-regulated. And there are some really interesting things that we have time we can talk about that we're looking for here. But of course, as I wanted to expand out earlier, right, I was really interested in looking at more than just Caulobacter. And as I said, one of the big ideas I've had recently is to start doing comparative analysis between a lot of these alpha proteobacteria and their phage, to start asking whether or not the mechanisms between Caulobacter's φCbK are similar to the ones, for instance, from Agrobacterium. Are these XREs always regulating similar types of genes, or are they facilitating sort of unique diverged functions from each of them? And sort of another interesting thing about these XREs is we know the numbers of them vary wildly across these species. Things like a lot of, for instance, the intracellular pathogens like Brucella and Bartonella only have one copy of these XREs. And obviously the one that we talked about today, Caulobacter crescentus, has 4 copies. But we know that there are species of Caulobacter that have up to 12. And several of these, for instance, Magnetospirillum has up to 8 copies. And so you can see a large radiation in a lot of these lineages. So one of the big questions is to start asking why we have these radiations. Do we see neofunctionalization, subfunctionalization of these different paralogs, or is this always sort of like ecological paralogs where you just always want it around in different conditions? To start looking at this. And so, of course, I want to give my acknowledgments to my mentors, everyone here, our funding sources, and I'll take any questions.
Okay, questions for Maeve, and friendly reminder, please say your name before your question.
Hannah, University of Colorado Boulder. Very cool. I was wondering, so phage can go lytic or lysogenic, and I'm always just amazed when transcriptional things can do anything for a lytic phage because it's so fast.
Yeah.
Do you think this could have any role when it comes to lysogenic, or have you
so
go lysogenic?
Yes. So there was a recent paper that came out. We had never been able to identify a lysogen for this φCbK. We don't think it can form a lysogen. There are some pieces that are required that are sort of missing, that it doesn't have, I think, a certain recombinase for it. But one of the things that I've always been really interested in is the fact that, like, it's like in your talk, most of what we do, it's not insignificant, but it's not real to what they're experiencing. Oftentimes we find these bacteria in a lot of oligotrophic environments, so there's not many nutrients. They're not growing fast. We also do find them a lot in like soil conditions. And so one of the questions is, we know, for instance, that cell lysis pathway is primarily induced in stationary phase. And we know that oftentimes viruses can't replicate during stationary phase because the host just doesn't have enough stuff to do it with. Mm-hmm. And so this may be one way that, sort of in this term of like lytic deferment, of like preventing the cell from lysing and waiting until things get better. At least that's one of my theories currently, as I'm hoping to test.
Cool. Thank you.
Hi, Zoe from the University of Utah. I was struck by the similarity between the phage XRE and the Caulobacter ones, and also the fact that you see it in a bunch of different Caulobacter phage.
Yeah.
Do you think that was a single acquisition from the host genome? Do you know anything about the evolutionary
Yeah, so
history?
let's see if I have that slide. Nope. Okay. No, so we've done pangenomics for these phi-CBK phage, and it breaks the different XREs from the different species, the different phi-CBK-like phage. So all of these are members of this phi-CBK-like group for these Caulobacter phages, but they do separate out into their own distinct groups. One of the things that we've always been interested in was, for instance, if you do a comparison of like Caulobacter to its Caulobacter's phage and look at their XREs versus like Agrobacterium and the Agrobacterium XREs, they're always more similar to each other than they are across species. Mm-hmm. And so we know that these infect several different types of Caulobacters, and obviously, like I said before, like some of them have up to 12 copies. And so we've never checked though to see whether or not they sort of fall into certain species versus crescentus, which we use as like our lab work dog. But yeah, that was sort of one of the interesting things. We think they've co-opted them from a lot of their hosts rather than from a single event. Okay. But for some of these that do fall into very closely related ones, we do think that was from a single event.
Yeah. Katie, University of Utah. I think this is super cool, and I have so many questions, but I'll just ask one. Well, first, this is a naive question. What is the end game for the phage infection here? I think somebody asked a little bit about that, but like, does the bacterial cell ultimately lyse?
It does.
Okay.
Yeah, they have several. They produce a lot of these things that basically pop the cell open.
Okay. I just think it's really interesting that. So I'm a mammalian immunologist mostly, and one of the clever ways that the mammalian immune system, with probably a lot of other immune systems, has gotten around the blocking of cell death is to just come up with other ways to kill off those cells. And so I was wondering if you think there's any chance that there is some other pathway that could, how do I ask this question? Like, is there another way for the bacterial cells to kill themselves that you've seen in looking at these other species?
Absolutely. Okay. So there's a lot of different systems. So there's like these CBAS systems that are quite prevalent that alter nucleotides and stuff. There's some of this ZOR system, which often it's thought to pop the cell open. But one of the big ones that's gained a lot of sort of traction recently is that there's a lot of toxin-antitoxin systems that are prevalent in bacteria. And it turns out a lot of them are really important for phage lifestyles. Essentially knocking themselves out and being again, cell death style things. Turns out this XRE also inhibits a lot of these toxin-antitoxin systems. And so that's sort of one of the feelings of I think the benefit for the phage in this case is that it's got a gene that suddenly hit a bunch of downstream targets that were very useful. And I think basically the host doesn't want to turn on either because they're not great unless you're in a bad situation.
Yeah, that's crazy. Thank you.
Hi, Sumra from University of Cambridge. I know nothing about bacteria or anything remote. So I was very interested in the relationship between inducing biofilm formation and how that's beneficial for the phage. I was curious if all these different species do they rely on forming biofilms, and do you see differences in terms of
yes,
whether
so it's funny. Some of them, for instance, Rhizobium legumicorum here, one of their big XREs actually does the opposite. It acts as an inhibitor of biofilms by repressing the adhesins. And one of the ideas that I've thrown around is that their lifestyle is very different from Caulobacter's. Caulobacter sticks to a surface and then just chills there and replicates. Whereas these ones down here often have to bind to a root and then get in. And one of the ideas is during that process, if the phage can't get in with it, then it may not be able to go to it. And so in these cases where it's acquiring these XREs, is that a case where it's trying to stop biofilm formation because it doesn't want to go into that life cycle where it's not ideal for replication? just one thought I've had, but that's sort of,
Awesome. Thanks.
they've differed, yeah.
Oh, sorry. I got it. Like, it's so easy.
Faster than me. Okay, so you sort of alluded to this on the last question, but I'm curious what the transcription factors do natively. So, like, you have this RNA-seq data from the XRE phage.
Yeah.
How does that compare to the XRE 1, 2, 3, 4?
Almost. So we have we have data sets for XRE 1 and 2, and it pretty much almost entirely overlaps. Okay. It's sort of the interesting thing of like I almost expected it not to a bit, because I was like, okay, it's got to have done something, like it's had to specialize it in some way. I will say that if we look, this is a comparison of the ChIP-seq about the number of peaks that are overlapping, and this is the one I was talking about where 99% of the peaks overlap. But it turns out that XRE 1, 2, 3, and 4 bind an enormous number of sites. One binds up to 1,200. And so one thing we've been interested in is that the phage one only binds about 170. And so it might have whittled down the amount of things that it's wanting to regulate in that sort of capacity. But they almost always tend to be the ones that are most highly regulated by the other ones, which is common amongst transcriptional paralogs. They often have this very core set of things that they regulate, and then like little babies on the sides that they sort of tinker with.
I guess, do you think that because of your data where you knock them down and you see loss of infectivity, do you think there's like cooperativity? Are these like dimerizing, and that's how you're getting some interaction between XRE phage?
We do know, so this is just,
Cool.
I always teach the students when I teach them how to present to let people ask questions that you already know they're going to ask. And so you always have it ready. We did a lot of homomeric interactions because most transcription factors are dimers or tetramers, but mostly. Turns out that all of our host XREs interact together, and our phage XRE interacts with itself and 3 and 4, and actually interacts with 3 better than it with itself. And 3 was actually identified in a screen a while back. So we think that 3 might be the most relevant one. In truth, when you look at sequence similarity, 3 and the XRE phage are more similar to each other than they are to this group of 1, 2, 1 and 2, which tend to be separate and funny.
Cool.
Thank you.
All right, I saw more hands, but I'm sorry. We're out of time. Please catch up with Maeve at one of the breaks or meals.